What is a neutrophil? And that's a question that may seem really obvious to a lot of you, but we talk about neutrophils as a really hallmark cell of the immune system, but sometimes we have to go back to basics just to make sure we understand the fundamentals to then build upwards about it. So this talk is very, very brief, but it's to go through the very beginnings of what a neutrophil is and how it fits in to the rest of the cells we know of the immune system. So some really clear pointers, first of all. The neutrophil is an innate immune cell. And it is a real archetypal cell of this part of the immune system. And it has one major job, and its major job is to carry out what's called phagocytosis. We're not going to go through phagocytosis in this talk because I want it to be short and brief and basic, and we will do that in follow on talks. Phagocytosis, what does that mean? It means that you are going to go in and attack anything that's threatening. Threatening being defined by something that's causing damage to you or something also that is non-self. So you're going to go in and round up, attack, and destroy those particular things. So a neutrophil is a real enforcer, it's a fighter. It's one of my favorite cells because it can also be a little bit crazy when it does this, and that can cause what we call immunopathology. It can provide a lot of collateral damage, unfortunately, in its process. But without them, we're in deep trouble. And again, as time goes on in this channel, we will start to look at clinical cases where we start to look at the ramifications of not having neutrophils as well. So let's go in and start looking at what we have. So the neutrophil is nearly always shown pictorially like this, okay, and so you understand why it's drawn like this in a cartoon fashion. It's often because what we actually have is a microscope picture here that you can see that shows what they look like when you produce a blood film. Okay, so here you can see a magnification over 1,000 times magnified, and you can see in here that we've got a very similar shape, and we've got two distinct colors. So the lighter purple that you can see here is the cytoplasm. This is the water that's inside the cellular membrane, and the dark purple is the cell nucleus. And the cell nucleus does not look like the traditional cell nucleus of a cell that we're used to seeing, and we're going to cover that now. And just so you're clear, these cells aren't really purple and pink and lilac. They're purpley pink and lilac because that's how we stain them with particular chemicals and dyes to be able to contrast and show the definition between different components in the cells. So these cells do not normally look like this, just so we're clear. They're actually fairly transparent. They're just like little water bubbles containing proteins inside them, and we color them up so we can see them clearly. Okay, and when we do the pictorial cartoons, we often replicate what we've seen under the microscope, and that's the history behind why they're drawn in these color schemes. So if we break down that picture that we have in the cartoon, why do we only include really three components when we're talking about a neutrophil? And that's because they kind of represent the three things that we know are most important about the neutrophil. So if we cover those, we've got the nucleus, we've got the granules, and we've got the cell body, which includes the cytoplasm and the cell membrane. So if we look at the nucleus first, I said before, it's atypical. It doesn't look like a traditional nucleus, which would be a big round blob. And if you look up in the total immunology image, you'll be able to see there that we've got a big traditional nuclei in three of those cells in the image there. So we call this a tri-lobed nucleus because it normally has effectively three bulbs to it, and then they're joined together by strands of material to put them together. And this is still where all the DNA exists for the cell. Remember, that's the point of a nucleus in a cell, is to contain all the nuclear material, so all the genetics, to effectively be a blueprint for you to be able to build another cell if you needed to, or for any of the proteins that cell utilizes to be made from. Why is it trilobed? Why doesn't it look like that? And there's no super clear answer about that, but there is some work out there to suggest that the reason it does this is because it gives the cell more flexibility. Remember, I said this is like a bag of water, effectively. And so what that water balloon wants to do is be able to squeeze in the sense of a neutrophil. It lives in the blood, but it wants to be able to get out of the blood to go to where the problem is in the body. So we call that trafficking. And it wants to move and migrate over to where the infection is in the tissue so that it can start to fight on your behalf. And in order to do that, it has to squeeze through some very, very tight structures. So if you imagine someone trying to go caving, and they're trying to go through some very tight cracks in rock faces, you have to contort your body to be able to get through. And it would make your life a hell of a lot easier if you had a lot of flexibility in your body so that you didn't get caught up. So it doesn't contain any bones, but the nucleus can be quite large and it can have a level of solidity to it. So if you develop a nucleus that is a little bit more hinged and enables you to be able to do that, you are much more adept at being able to squeeze through and out from one site to another site. Now, the posh word for being able to squeeze through is called diapedesis. And again, we're going to cover that a little bit more in another talk at another time. So if we move on to the granules then, the granules are these little blebs. You can see they're all the different sizes and shapes. And the reality is that there are lots and lots of vesicles in a neutrophil. And it's one of the characteristics of it that we're used to seeing is it has lots of these in. What's inside them? They contain enzymes or particular protein complexes that allow us to do particular chemical reactions. So it may contain enzymes that are digestive, that will start to break down anything that is near it, particularly if it's going to eat it. And phagocytosis really is a posh word for ingesting and engulfing something so you can break it down inside. So it's the closest thing we can get to a cell eating something else. And it needs to have enzymes which are smaller proteins to help break down the structure of whatever it's eaten. It can also produce these toxic compounds, and one of the compounds it can make is hydrogen peroxide. So as well as it being good to bleach hair, it's a very reactive compound that wants to give off oxygen-free radicals. And oxygen-free radicals, as anyone who's anti-smoking will tell you, are bad. They damage, they stick onto things and change chemical structures very spontaneously and very unregulated. And so it can cause a lot of damage in the long run. So these granules are the weapons of our neutrophil for it to be able to hurt and damage threats that may be coming to you. The final part that we have to look at is the nucleus. So if we then start to look at the nucleus, the cell body and the cytoplasm, excuse me. So when we look at the cell membrane and the cytoplasm is just a lot of fluid, and predominantly it's pH 7 type water that is osmotically balanced for the cell, and it's going to be where a lot of things can move around inside as a fluid. The cell membrane is very important to a neutrophil because it's actually the site of where it's going to be able to sense the environment around it. It needs to be able to know if there's a threat there. It needs to be able to know if it's going in the right direction towards a threat. And in order to do that, it has to use receptors. And remember, receptors are simply protein constructs that have a particular shape on them. One great example of a receptor that a neutrophil uses is called a toll-like receptor. And it looks a little bit like this one here. And a toll-like receptor is designed to be able to recognize signature molecules that are produced by particular types of organisms, such as bacteria, fungi, and viruses, in order to be able to detect by a process of evidence elimination that says, if I can see this compound, it must mean that this sort of organism is here, and that's a bad thing. So we're going to talk about toll-like receptors again in more detail. This is an overview here. But what you need to understand is that your neutrophils are absolutely covered with these receptors all around them in order to be able to sense everything around them to know if there's danger there. And any triggering of these receptors will activate the inside of the cell and will make this neutrophil react in a manner that means it's going to try and protect you. And it's potentially going to move in the direction of increased concentration of stimulus by these receptors. And that's another posh word for us to bring up now, which is chemotaxis. So chemotaxis is going to be the thing that makes our cells migrate in the correct direction towards the threat that we see. So a neutrophil, where does it come from in the body? How is it made? Well, it's made in the bones. It's made in the bone marrow alongside all the other cells that are in the bloodstream. So including red blood cells and including platelets, we have the neutrophils. Now, neutrophils are actually part of what we would call the white cells. So they all come from one original cell, and that cell is called the hematopoietic stem cell. And the hematopoietic stem cell is our cell that that we know we can generate any type of white cell from after this. We have two particular types of offshoots from that original cell, and one is called a myeloid progenitor and one's called a lymphoid progenitor. And if we just concentrate on the myeloid progenitor, we get three particular types of cell that come off this. It's not the only cells that are made, but I want to concentrate on these three for this talk. that are all very similar in their makeup. And you can see from the pictorial representations of them that they all look very similar. Really, what the difference is that they have slightly different shaped nuclei, but they're still lobed. And the color of the granules are different because they represent the different types of chemicals that they carry inside them. Actually, if you look at these guys under the microscope, you will see that they look different if you use different dyes. So eosinophils, for instance, are called eosinophils because phil means to have a love or an attraction to. And what we have here is eosin is a type of dye that we use, which will dye those granules inside eosinophils very brightly, which makes them stand out when you're doing differential cell counting. These three cells are collectively known as the granulocytes, and you can see why, because they've got loads of granules inside them. You can also sometimes call them polymorphonuclear cells, so PMNs as well. Okay, so be aware, you might see that in literature occasionally. So how common are they in the blood? And that's the last thing I want to cover in here, is to get a feel for, we have all these different cells that are produced in the blood from the bone marrow into the blood, but are they all presented or represented in the same quantities in the blood? And the answer is no, definitely not. And what I've got here for you is a scaled proportion of representative amounts of cells. If we were to take the median full blood count range of a medium average healthy human adult, okay? And just to show you, these aren't the exact numbers, so don't go counting them on the slide, but it's just a representative proportionate size where I've done some maths to scale them across. But if you understand that a neutrophil is the most abundant white cell that we have in our body, okay, so there's the representative amount of neutrophils we might see in the blood of our healthy human. If I look at the representative amounts of the other cells we might see, you can see that eosinophils and basophils are nowhere near as many as we have of neutrophils. And if we look at other types of cells that we know are common when we look in the blood, so those are, for instance, to look at the monocytes, which we can also think of as macrophages, and there are subtle differences. Again, we will talk about that in a separate talk. And the other most important group, which is the lymphocytes, which are the archetypal cell type. It's an umbrella term for multiple different types of cell which are associated with adaptive immune system. You can see that there is a lot of those, still not as many as the neutrophils. So the neutrophils are very abundant, high concentrations, and their numbers can really fluctuate in an infection. In the early stages of an infection, we could see that number of neutrophils double or triple, and that would be a normal response in those situations. You have to also understand how long they live for. So we aren't just getting cluttered up with them because they're being produced and they don't die. They do die. And actually their half-life is very short. And it's the shortest amount of any of these cells. And it's approximately about 6 days. So every 6 days, 50% of your cells will have died that came out on day one. And so they have a high turnover, high production rate all of the time, and your bone marrow has to have a big reserve of them to be able to work. So hopefully that's been useful for you. Please remember to email if you have any problems, or put any comments, or subscribe or like the video if you like, and look out for more videos that come about the basics of our different cell types in the immune system. And thank you very much.